Hey, I'm Jake.
I'm
Ella.
And I'm Jack.
And this is our 7th grade bacteria research project. We're in Sideburns, and we are in Table E in our science class.
Our group is researching which sugars will help bacteria grow and which ones will not. We are researching this because we did an initial research that found out that bacteria eat sugars to grow. And these are our sub-questions, which are Jade is researching why does bacteria feed on these sugars. I am researching how bacteria metabolize them, and Ella is researching what's the difference between the sugars that we are researching, calorie-wise and otherwise.
So, our question is, how does the amount of calories in sugar help bacteria grow? We know bacteria uses sugar for energy, and when they take it in, it puts something out called fucose. But would that make it multiply across a plate of agar? And this question is interesting because if you made some cookies or if you were just eating sugar, wouldn't you like to know if there was a possibility of bacteria growing in it? So, here's our hypothesis. If there are more colonies of bacteria on the plate that have the sugars with more calories, then the bacteria will use the calories to multiply. This will prove that bacteria feeds on calories, and they are sometimes in more food with more in food with more calories, there will be more bacteria.
Okay, so this is the bibliographies here, and if you didn't know, a bibliography is basically where you are researching something and you take the source. So you take the source from the website, you put the author in, and basically you summarize what is in your reading, and then you put some facts that you have. So I took 2 that all of us did for each of us, and as you can see as I'm scrolling down, that all of them have the same thing: an author, a source, a summary of what they read, and some facts about it. Okay, so our group is researching different types of sugar and its effect on bacteria. We think the sugars that have more ingredients and more calories in them will have more bacteria on our plates. So we'll be testing raw sugar, powdered sugar, brown sugar, corn syrup, Splenda, and C&H. Okay, so on our first source, which is Science News magazine, we found out that bacteria takes in sugar and eats it and then puts out a byproduct called fucose. It helps your digestive system because it breaks down the foods, and it's just helpful for your body. So our independent and dependent variables are the sugars we're testing and the time in the incubator. In conclusion, the main idea of this experiment is what kinds of sugar will help bacteria grow the best on our plates. We think it'll be Splenda because it has the most calories and ingredients in it. The more ingredients you put in, the more processed, junky it is for you and obviously the bacteria. So, and another way you could take this experiment further is by taking one kind of sugar and testing different kinds of liquids to dissolve the sugar with, and then see how it affects bacteria growth.
All right, here's our expected graph. So, we expect that in the sugars with less calories, there will be less bacteria. And as it goes up, you can see it's going up at a slope here. It'll get progressively higher with the more calories you have.
This is our material section. This is where we list what we need, where to get it from, and why we need it, and a little bit about it, and what role it plays in the experiment. So here we're obviously our sugars, which are powdered sugar, raw sugar, corn sugar, C&H, a brand sugar, Splenda, and brown sugar. Measuring utensils for measuring out the water that we add and the amount of sugar. Plates or petri dishes from our science teacher Saber. Q-tips to act as the inoculating brush onto our plates. Water obviously to dissolve the sugars because we can't add it straight onto the petri dish, parafilm to seal off the plates when we put them onto the incubator, an incubator to grow the bacteria, and a computer to take pictures, data, and obviously the strain test. And a marker to mark the plates so we know what they are. And this is our procedure. It's where you list things to do to replicate this experiment. So as I scroll down, these are some of the pictures that we found. Choosing the sugars, choosing the plates, dissolving the sugars, and we are pouring the sugars out, dissolving the sugars, and then inoculating the plates, which is a big step. And then repeat it for all the other sugars, and this is how we do our negative control. It's basically steps 2 through 7 on our procedure, except using only powdered sugar, because we think it will grow the least bacteria. And the positive control, which is, again, steps 2 through 7, except only using the brown sugar. And then this is how we check the plates. After 2 days in the incubator, we count only the colonies down from the center and to the right. Then we just throw the plates out.
Yes, so here's how we streak. I was the streaker. So first you divide the plate up into thirds. If you can see, here's a third, and that's a third, that's a third, and that's a third. And so we start by dissolving the sugar, and then we go across in the top 6 times, and then to the right, bottom right, and then we go 6 strokes, and then across to the left and 6 strokes. And this area here is where we check.
Okay, so this is our data table where it shows all of our actual results. So for brown, which is trial 1, we had 0 colonies, trial 2, 0, trial 3, 12 complete colonies, so our average of colonies was 4. Raw was 1 colony for the first trial, 1 colony for the second, 0 colonies for the third trial, so that's 0.667. For CNH, it was 0, 0, and 0, so our average was 0. And for powdered, it was 12, 1, 0, and our average was 4.333. Splenda was 0, 1, 0, so our average was 0.333. Corn was 0, 0, 0, so our average was 0.
And here is our actual graph. So this was a real big surprise to us, because if you remember from the expected graphs, we expected it would kind of have a motion that went up, but this is just kind of completely random. See, it peaks at 3.2, but then it goes down for 3.752, which is a lot more calories. Well, not really, but a lot in terms of how much we're using. And then it went back up for 4, which we kind of expected, but 3.2 was still higher. And so we can draw that the amount of calories didn't affect the growth of bacteria.
And then these are our actual plates. This is trial 1, which are the 2 graphs in the first row. Trial 2, and our 2 trials. And then our third and final trial, you can see the bacteria growth on brown on the right, and a few specks for the others on the left and the right.
Okay, so here's our data analysis section. So we did, we kind of summarized basically all the data we collected. So CNH had 0, brown had 4, powdered had 4.33, raw had 0.667, corn had 0, Splenda had 0.333. And the third trial tends to have the least amount of growth that we have. So our negative control was raw, our positive was corn, and our control was powdered sugar. On trial 3, we had the outlier of brown sugar. During the first trial, powdered sugar, there was 12 colonies, as the others had 1 colony. So that was also an outlier that we had. So our constants was the amount of water we had, the way we mixed the sugar, and the amount of the sample we put on our plate. So this is basically it.
And so from this experiment, we can conclude that the amount of calories in sugar does not affect the amount of growth in bacteria. Thank you.
For watching.
Thank you. Bye.